results of his project. And Max Mann is a junior at Falmouth Academy. And he won the second place award at the South Regional Science Fair for his project Search for Marine Microbes that Metabolize Dissolved Organic Carbon. This year, Max worked not only with a biologist, but a chemist at Westphal Oceanographic Institution. I'm sure he has a lot of great results to share with us. So, Max, what was yours?
Thank you. The project that I spent my time on was revolved primarily around looking for a certain type of microorganism by testing several types of microorganisms that has the capacity to degrade or break down dissolved organic carbon in its normal metabolism. Now, the background on dissolved organic carbon is that its exact chemical structure is yet unknown to the scientific community and has been somewhat of a mystery for the past 90 years when scientists initially began to study the chemical composition of the ocean. Some of the structures are unknown, but they're believed to be some kind of polysaccharides created through the release of carbon by algal photosynthesis. This carbon is thought to represent the main carbon reservoir for microbial metabolism in the marine environment, and it can be split into about 3 functional groups of varying reactivity. The one I'd like to concentrate on is the one in the middle, which represents about half of the total concentration of dissolved organic carbon in the world's oceans, and it cycles on a seasonal to decadal time scale, so any amount that you see is only so old. This latter group, the approximately 40 micromolar concentration of non-reactive dissolved organic carbon, which isn't seen to be very prevalent in the carbon cycles in terms of biology, takes about 4,000 to 6,000 years to cycle through the ocean. And that'll come up in a minute. The importance of dissolved organic carbon research is in understanding precisely what microbial processes take place in the open ocean that are necessary for all the life on land and on the rest of this planet. So marine carbon cycling would be vastly improved by the knowledge of precisely what exactly this dissolved organic carbon is in terms of its molecular structure and what applications it has in terms of microbial life. The U.S. Department of Energy spent a lot of money in researching carbon dioxide sequestration, that it might be possible to find some way of taking carbon dioxide either from the atmosphere or directly from power generating facilities and fix it to a soluble form of carbon, such as the non-reactive form of DOC that's found in the world's oceans, such that it might be removed from the atmosphere without changing the pH of the oceans. The purpose of the project was to break down dissolved organic carbon by creating cultures for these bacteria such that later analysis could take place of the similar products of the dissolved organic carbon. So in order to culture these organisms that will be discussed later in dissolved organic carbon, a medium was created with this carbon as the sole carbon source, such that any organisms that grew could be established to have grown on the carbon and to have metabolized it such that it might be later chemically analyzed. Then to obtain isolates such that a sort of reverse engineering could take place to obtain clues as to DOC's structure. This reverse engineering would take place by analyzing the proteins that are used in creating the enzymes necessary to break down this compound. The experimental design revolved around taking the isolates of organisms that are known to degrade complex polysaccharides and other materials and to take enrichment cultures from the environment and determine which sets or whether all of the sets could degrade the DOC. Cell counts were used to determine whether or not growth was effective and whether to continue or discontinue cell lines. Total organic carbon measurements were taken initially and every week thereafter so as to get some measure as to the change in carbon levels due to microbial activity. Finally, culturing on DOC medium agar was used to isolate and ensure the purity of cell lines. The presence of other bacteria or organisms that were not wanted would be able to be visible simply by colony morphology. The inoculants used included isolates such as Cytophaga-like bacteria. There were 2 varieties with some names that they're only recently discovered, and they're currently called Rus and Tangerine. The Pteridinobacter turnerae is an organism that was isolated from the gland of Deshays in wood-boring mollusks. This is the organism that is responsible for cellulose degradation in these organisms. The Microbulbifer hydrolyticus is another organism that is capable of degrading very complex polysaccharides, among other interesting materials such as chitin and tween 80. Enrichment cultures were created from local sources, such as Shiverix Pond, a pond near a sewage runoff area, Trunk River, an estuary to represent both freshwater and marine microbial activity, and Weeks Farm, one of my favorites, the manure pile, because there's so much microbial activity there. This graph of cell counts represents the number of cells noted in the Trunk River control and the Trunk River 10x DOC. When these cultures were created, 10 times the normal concentration, the ambient concentration you'd find in open ocean, was used so that changes in the DOC could be more readily apparent. You'll note that the set with more dissolved organic carbon grows about twice as much as the set that has none. But it was initially confusing that the set that was intended to have absolutely no carbon in it, this control has no dissolved organic carbon, ferric citrate was avoided because that would have caused extra carbon to be added, and has no other food source. You still see almost the exact same rise in growth and then falling off, just as though it were the same exponential growth. That same trend is observed in the second graph of the Shiverix pond enrichment culture growth with different DOC concentrations, where the blue line is the control, the lighter blue line is the 10 times concentration, and the green line is 20 times concentration. You'll note that in the green line, the growth is even greater than in the other two, but the same general trend is observed. Interestingly, the first day, on the bottom you'll note that those are hours after inoculation, and the first line approximately relates to 1 day. In the first day of growth, all of the cultures were observed to have the same amount of growth, which is completely baffling. We'll get to exactly why that might be later. Just a quick note on this calibration graph for the TOC-A, or the Total Organic Carbon Analyzer. This is the calibration graph, and it shows that there's an R-squared value of 0.9993, representing a high degree of accuracy in all the measurements that are taken in this graph. It's important to note when viewing this information that the bars shown are the total organic carbon present. So any amount that was there was measured. If the dissolved organic carbon was degraded somewhat but remained in solution as opposed to exiting solution by complete oxidation to an inorganic carbon such as carbon dioxide, then there would show no decrease. In similar tests like this one, decreases of 99 or more % are common as the micromolar concentration goes from as many as 800 micromolar down to 1 micromolar. That's true for tests of glucose and many other tests involving carbon usage. Here we see almost no change at all, if not some increase. In fact, every set shows an increase of at least 5%. And that was initially completely baffling, because there should be no reason why there would be an increase in concentration of carbon, unless one considers the experimental design, in which cultures were inoculated and allowed to incubate at approximately 30 degrees Celsius for several weeks. At a temperature of 70 degrees Celsius, with the amount of surface area of the liquid, it was calculated and weighed in a separate experiment that approximately half a mil of culture evaporated per day. And so that those changes over time add up to decreases in overall amount of solvent and an increase in concentration of solute, or in this case, the total organic carbon. So that would explain some of these increases in dissolved organic carbon. The only set that shows a decrease, you may have noticed, is this week's 10x set over here, which shows in its first week a decrease of approximately 11%. That decrease may be attributed to significant decrease in the dissolved organic carbon that was present, but it also may represent the metabolism of a more labile contaminant, one that's more readily accessible by microorganisms. This is definitely possible because the week's set was from the week's farm, or the manure pile. And manure is known to have a very high number of carbon compounds. And so even after 3 transfers, it's definitely possible that about 150 micromolar concentration could have been from the manure. Evidence for this theory exists in the second week. Note that the initial week is in light blue, the second week, in blue, and the third week in purple. Between the second and third week, the concentration increased, indicating that indeed, once this labile carbon was used, there existed nothing left. So the analysis of this, well, we said earlier that the increases in concentration could easily have been due to evaporation. In fact, it was shown the mechanism by which this could be true. Possible labile contaminant in the week's farm culture would explain that initial decrease, but is not necessarily what we're seeing. The presence of ammonia-oxidizing bacteria may be able to explain that conundrum of the initial cell counts that were shown to increase even where no carbon was present. These ammonia-oxidizing bacteria would take the ammonia from the ammonium chloride that was added as a chloride supplement in culture creation, and they would oxidize it to nitrite. So a simple nitrite test was performed, a colorimetric test to determine whether or not there existed any nitrites. That test proved negative, but yet it still remains that there existed nitrates, further oxidized from nitrites by other symbiotic bacteria or bacteria working in conjunction. So further testing would be needed to determine whether or not this ammonia-oxidizing bacteria represents a possible candidate for change. The experiment may represent successful partial metabolism of dissolved organic carbon and may still be useful to scientific assessment. In this project, only the cultures of cells were created and the isolates begun to be isolated. And so the metabolism of DOC, the extent to which this DOC may have been chemically simplified by the addition of these organisms is yet unknown. So it's important to continue by analyzing this DOC. An important thing to note is that dissolved organic carbon is found in concentrations of approximately 80 micromolar around the world in lakes, ponds, rivers, open ocean, just about everywhere about the same concentration. And it was a general scientific belief that dissolved organic carbon self-regulates because an increase in concentration would cause it to be more worthwhile for organisms to degrade it and cause it to decrease in concentration as a result. This project did not show that because, as shown in the previous slide, even with concentrations of 10 times and 20 times ambient seawater volume, and with the same organisms that are found in seawater, there was shown to be little or no decrease. Finally, to continue the search, further culturing is necessary to determine whether microbes can fully oxidize DOC. It's possible that the inoculants used did not contain precisely the right bacteria. It's really a pot shot of sorts. Also, the presence of nitrate should be determined because it's important to find some sort of an explanation for those confusing cell counts. The post-culture DOC, or that dissolved organic carbon which remains after the application of organisms, should be chemically analyzed to determine whether or not any partial decomposition existed. And finally, secondary carbon sources should be investigated because it's possible that dissolved organic carbon metabolism in the open ocean is only made available by the presence of secondary carbon sources that act as a sort of way of allowing organisms that wouldn't get their full energy requirement from DOC alone to subsist. Sort of like eating pizza with cabbage because you wouldn't want to eat the cabbage alone. I'd like to acknowledge Dr. John Waterbury, my mentor, Dr. Dr. Daniel Rapetta, he's not on this list, who was the geochemist with whom I worked, and Dr. James Johnson.
Time for a couple of questions for Max, and then Max will be presenting his board after all of the talks are given, so you can talk to him in more detail. Does anybody have some quick questions?
Question, did you aerate your cultures?
Yes. These cultures were grown in, well, they weren't, they were grown in air, but they weren't, no, they weren't aerated. It is known that the organisms that we tried were mainly aerobic bacteria.
Did you consider the loss of organic by vaporization?
The loss of organics by vaporization.
Gaseous state.
To explain what? A decrease in concentration would of carbon would result from the vaporization of carbon, but increases were found. Certainly it's possible that carbon's evaporated, but what is known is the evaporation of the solvent, of the water in culture.
In your analysis page, you show several things. One is the concentration as a result of evaporation. Several in there. Which one of those do you think is the most
The most prevalent?
Yeah.
I believe the most prevalent is the increases in concentration due to evaporation. It's certainly a large factor when you consider that each time carbon measurements were taken, the amount of material that had to be removed from the culturing flask was approximately 30 mils, or 30% of the available culture, leaving behind a much smaller amount, such that a same rate of evaporation, which was observed in later tests, would result in even higher percentages, up to 9%, which would certainly explain increases as shown in the 20x set, which was to the far right.
Thank you.
More questions? Go ahead.
How did you measure your organic carbon?
The dissolved organic carbon was measured with a total organic carbon analyzer, an auto-sampling machine that uses high-temperature catalytic oxidation in order to burn the carbon that's present in small injection samples, and then measure with with an ionization detector exactly how much carbon there was. That carbon, that measurement calibration I was showing you before, it's important to note that even though we're using very high concentrations, the injection size is automatically adjusted for the high concentrations so that the amount that's measured is always falling within the calibration range of the instrumentation.
Thanks, Max. Second speaker this afternoon is Kevin Lynn from Falmouth High School.
Put that on your belt. I'll put this on.
First place award at Falmouth High School, a second place award at the regional fair, Pleasant Fair, and the title of his talk this afternoon is Evolution of Marine Cyanobacteria from the Red Sea.
All right. Okay. My research focused around one particular organism known as Prochlorococcus. This organism, as my title suggested, is a cyanobacteria, or a blue-green algae. And the uniqueness of this organism comes primarily in its size and its range. So this is a photosynthetic prokaryote, meaning it's a single-celled organism which uses light as an energy source. With a diameter of just 0.5 to 0.7 micrometers, it is the smallest known photosynthetic organism. It lives anywhere from 40 degrees north to 40 degrees south of the equator. And within the water column in these areas, it can live anywhere from the surface to 200 meters deep. This wide range of habitats makes it the most abundant photosynthetic organism in the oceans, and therefore it accounts for 2/3 of the ocean's photosynthetic processes. So this organism is significant because of its small size as a food source and also as a source of photosynthesis for ocean ecosystems. This map just shows a range of where Prochlorococcus can be found, anywhere from the Indian Ocean to the Pacific Ocean to Atlantic Ocean, or to the Mediterranean Sea even. And the particular strain of Prochlorococcus that I analyzed, strain 83809, was isolated from the Red Sea. The Red Sea is Gulf of Aqaba. And this gulf is located approximately 28 degrees north of the equator. But the uniqueness of this environment is the annual mixing and stratification cycles that occur. Between the months of April and October, nitrogen compounds fall from the top of the water column down toward the bottom, leading to nitrogen stress in the upper photic layers where sunlight penetrates the seawater, and therefore organisms must adapt somehow to find alternate sources of nitrogen. So the purpose of this project was to determine if the organism Prochlorococcus strain 83809 from the Red Sea's Gulf of Aqaba has adapted to the unique environmental conditions of nitrogen stress in the Gulf, and how it therefore differs from other strains of Prochlorococcus found worldwide. So to do this research, I first sequenced and assembled the DNA from Prochlorococcus strain 83809. I next determined the functions of the genes in 83809 to determine how the organism interacts with its environment. And I finally compared the DNA from 83809 to other Prochlorococcus strains to determine how it has evolved. So the first part was a sequence and assembly procedure. Using PCR-based methods, or polymerase chain reaction, I began with a glycerol stock containing plasmids of 83809 DNA, and I amplified these plasmids and used a capillary sequencing machine to obtain the DNA sequences of 83809 in the form of the 4 DNA bases A, T, G, and C. And I assembled these sequences using computer software, as well as identify genes in these DNA sequences based on universal start and stop codons. So these are just 2 pictures from my labs. All right. So from these sequencing and assembly procedures, I obtained 62,000 or so DNA base pairs from the sample of 83809 DNA that I was given. And this DNA was assembled into 28 contigs, or 28 pieces of DNA, because connecting sequences were not obtained through this set of procedures. And within these 62,000 DNA bases, I was able to identify 67 open reading frames, or ORFs, which correspond to possible protein coding genes. So these sections of DNA are the parts that actually have a function in the organism. This diagram, or this picture, just shows the plasmid preparations, the DNA samples that before putting into the capillary sequencer. And this next picture shows the trace files, which were the raw data output from the capillary sequencer. The different colored waves correspond to the 4 bases A, T, G, and C, and using computer software, those were translated to determine what the DNA sequences of 83809 were. So with the 67 open reading frames already identified and the DNA sequences obtained, I proceeded to a functional analysis of this DNA to determine what the organism actually does. Using the NCBI database, the National Center for Biotechnology Information, I looked for matches between 83809 DNA and the DNA of other already known organisms. And these matches were examined for E-value, percent identity, function, and organism of origin. And based on high significance matches between 83809 DNA and the DNA from other organisms, I was able to determine what the functions of the genes in 83809 are. And from these procedures, I was able to find functional assignments for all 67 ORFs. In the database, approximately 108 matches were found for each gene that I plugged in. 67 of the open reading frames from 83809 DNA had the high significance matches to other Prochlorococcus strains, which makes sense considering it is a Prochlorococcus organism. However, 3 of the open reading frames, or 4% of them, showed the most significant matches to other organisms that were not Prochlorococcus, showing that there is some genetic diversity between the Prochlorococcus in the Red Sea and other Prochlorococcus strains. Finally, no functional assignments were found related to nitrogen assimilation or acquisition. This was a major point of interest, considering that the nitrogen stress conditions in the Red Sea can make it seem like the organisms would need some kind of genetic mechanism to deal with nitrogen stress between April and October. The fact that no functional assignments were found related to nitrogen assimilation and acquisition is a point that I will discuss later. This graph just shows the high significant matches for the Prochlorococcus A380C9 genes. As you can see, the majority of them come from other Prochlorococcus strains, such as Prochlorococcus marinus, Hotum 8F9 on the right, the Prochlorococcus strain AS9601 in red, and MIT 9312 in white. The green section of the graph shows the small percentage of organisms which were not Prochlorococcus. So having obtained these functional assignments for the A380C9 genes and knowing what the DNA of the organism is used for in the organism in the A380C9, proceeded to a phylogenetic analysis. I identified an organism that had many high significance matches to the A380C9 genes in part 2. I obtained the genomic sequences, annotated genomic sequences, for this chosen organism. I used the organism as a scaffold to align the A380C9 DNA and genes to determine how closely related the two organisms were, the Prochlorococcus A380C9 and the other Prochlorococcus strain that I chose. And based on the similarities and differences between the two different Prochlorococcus DNA sequences, I tried to determine some kind of evolutionary relationship between the two. So to go about these procedures, I literally cut out pieces of paper with the DNA of these two organisms. This first bar shows the DNA, the annotated gene sequence from Prochlorococcus strain Hotum 8F9. The blue just shows the DNA, while the red arrows indicate the gene sequences. And I took the 28 contigs from my DNA sample A380C9 and lined them up so that corresponding genes line up vertically. And as you can see with these yellow boxes, in the vertical columns you see that the similar genes line up, also with the same order. On the box to the right, you can see the sucrose phosphate synthase followed by a mRNA gene in the same order in both the Hotum 8F9 and the A380C9. So based on these similarities, I was trying to determine how they're related evolutionarily. So 37 of my open reading frames from A380C9 exhibited similar gene sequences to those in Hotum 8F9. So there are 37 out of 67 that matched up. But due to the overlaps, as you saw with the sucrose phosphate synthase, the number of gene sequences was reduced to 30. The number of unique gene sequences reduced to 30 from the original 67. 11 unique open reading frames showed no overlaps with the Hotum 8F9, showing that there is genetic diversity between the two organisms. 3 contigs were found to be completely unrelated to the Hotum 8F9 genome. The functional assignments did not match up at all. And 3 total genes were most significantly matched to other organisms, as I mentioned in part 2, the Shigella, Maricolis, and Melstonia, which are not Prochlorococcus organisms, but they are bacterial species. So from the results of these 3 steps, I found that Prochlorococcus, first of all, is composed of 30 unique open reading frames, which correspond to gene sequences. So in the section of DNA that I analyzed, the 62,000 base pairs, there are 30 protein coding genes. Based on the 96% of open reading frames identified from other Prochlorococcus species, I found that the A380C9 cyanobacteria is very closely related to other Prochlorococcus organisms. Thirdly, the 3 gene sequences identified from other organisms show that the Prochlorococcus A380C9 is is logically not identical to the other Prochlorococcus strains. There is some genetic diversity between the organisms. However, this was apparently not due to the nitrogen stress conditions of the Red Sea, which I originally expected. This lack of nitrogen acquisition and assimilation genes suggests that A380C9 represents an organism incapable of extensive nitrogen stress responses. To make this conclusion, it's important to understand that the section of DNA that I sequenced contained a gene known as COB-A, and in many other Prochlorococcus, Inoculants species, other cyanobacteria, this COB-A gene signifies an area of the genome where a whole suite of nitrogen acquisition and assimilation genes are usually present. So provided that these genes have not translocated to other parts of the genome, according to my project, this A380C9 organism does not have those genes present. And finally, Prochlorococcus A380C9 is likely more genetically complex than the uncultured Hautum 8F9 organism, as suggested by the 11 unique open reading frames that did not overlap between the organisms. However, these are both subjects of future research. The genotypic variation between Prochlorococcus A380C9 and Hautum 8F9 and the suggested genetic complexity can only be confirmed if the entire genomes of these two organisms are sequenced and the number of genes is counted in each. And finally, the study of A380C9's adaptation to the condition of nitrogen stress in the Red Sea through further genetic studies and also through laboratory experimentation to see how the organisms actually react to nitrogen stress conditions is definitely a future investigation. And I'd like to acknowledge Drs. Mitchell Sogan and Hillary Morrison of the Marine Biological Laboratory right down the road where I did my research, Dr. Anton Post from the Inter-University Institute for Marine Science for providing the samples and guidance in my project, Ms. Christine Brothers from Falmouth High School for supporting my work throughout high school, and the Oceans Alive Hui Sea Grant Program for allowing me to present my research. Thank you.
The genetic variation between humans is extremely small, but the genetic variation you had was, you said 96% the same.
Mhm.
So does that represent a small amount, much less than you expected, or more than you expected, or exactly what you expected?
I think it was about what I expected. I was just looking for some genetic variation based on these nitrogen acquisition and assimilation genes, which I would expect wouldn't be found in other Prochlorococcus strains in areas where nitrogen stress isn't a problem for them. So I was really looking to see how the genetic variation based on these nitrogen genes contributed to the genomic variation.
Kevin will be presenting his board. Our next speaker this afternoon is Neil Forrester, and Neil presented a project 2 years ago at Oceans Alive, so again, he's a veteran of these presentations. Neil received a first place award at Falmouth High School and a first place award at the South Shore Regional Fair, and he's off to Albuquerque, New Mexico to go to the International Science Fair in weeks. And Neil will be talking about his project, differentiating random sequences with a genetic algorithm.
So, my project, as you can see, is entitled Differentiating Random Sequences with a Genetic Algorithm. So, first of all, the random sequences of numbers are very important when you're trying to encrypt a message for security on the internet or in any other environment where you need to use a secret code. So, there are several common ways of generating random numbers. The most commonly used ways when playing games are dice or flipping coins, but for cryptographic purposes that's not exactly fast. So most people use either a radioactive source with a Geiger counter hooked up to a computer and extract randomness from that, or they have a CCD chip out of a digital camera in the dark, and the amplification there will cause good random data to come out of that. But unfortunately, most people don't have such devices hooked up to their computers, so they need another way to come up with random sequences that they can use to generate keys that they can encrypt their messages with. So other people use what are called pseudorandom number generators. And that's a computer program that is working to create a sequence of numbers which is not truly random, but is almost random. A computer cannot create a truly random sequence because it's a computational machine. It cannot produce anything that's not the result of math. But there are mathematical functions which can produce numbers that are close to random. A linear congruential generator, for example, is a fairly poor random number generator, but it's very easy to implement. Basically, it takes a number in the sequence, multiplies it by a constant, adds a constant, and then divides by another constant and takes the remainder to get the next number in the sequence. But like any other pseudorandom number generator, John von Neumann, one of the pioneers in the field, said that anyone who considers arithmetic methods of producing random digits is, of course, in a state of sin, meaning that you can't do that. It doesn't work, and you cannot generate real random data with math, but you can get close. So in order to determine if a pseudorandom number generator is suitable for cryptography or for encrypting a message, you run some tests on it. And these tests include the chi-square test, the equidistribution test, and some others. And what they do is they check the statistical properties of the sequence of random numbers produced. And they determine if it has various characteristics which make it non-random, such as having one number appear more often than another. However, I decided that these tests were not entirely adequate because they would only, each one individually, just tested for a specific quality that a random sequence would have that would make it actually not random. So I decided to apply a computer programming technique called a genetic algorithm. Now, a genetic algorithm is a method of optimizing a solution to a complex problem. And the way it works is it randomly generates a bunch of solutions, possible solutions to the problem, which can be represented mathematically as a bunch of numbers arranged in some way. And each possible solution is called a genome, and it randomly creates a bunch of these in the population initialization stage. Then it moves on to the objective function. The objective function will take these genomes, these random groups of numbers that represent possible solutions, and it will decide how good they are and produce an objective score. And this objective score will range over a certain range of numbers, and a higher or lower objective score, depending on how you define it, will decide how good or bad a certain genome is. Then it moves on to the death phase, where it will eliminate from the population those genomes which scored poorly. Then it will take the best genomes from the population, and it will breed them by taking pieces of their genomes and mixing them up into different combinations to produce new genomes, which are hopefully better. And then it will occasionally mutate one of these children by changing its internal structure or swapping some numbers in and out. And then it will go back to the objective function after reinserting these children into the population. So you end up with a new batch of genomes which need to be scored, and the whole cycle repeats. Each cycle is called a generation. And after many generations, you end up with a solution to the problem that's better than anything that you probably would be able to come up with on your own, assuming the problem isn't very easy. So the primary goal of this project was to use a genetic algorithm to come up with methods, algorithms that could differentiate between random sequences from two different sources. And each computer program that was being optimized by the genetic algorithm would take in a sequence of random numbers, and it would produce a value. And then this value would be decided if it was above or below a certain value. And if it was above, then it would be declared to come from one source. Or if it's below, it's declared to come from the other source. And the accuracy of this and refining the accuracy of these programs that are being developed is the goal of this, so that you can end up with a way to reliably differentiate between two sources of randomness. And if you can take a bunch of sequences of random numbers and sort them according to where they came from, then that enables you to do two things. One, if it's possible to do that, then you know that one of those two sources of randomness is not actually random, and it has some quality to it which makes it so that its sequences can be told apart from real random sequences. And the other thing you can do once you've got all these programs saved from a bunch of runs through the genetic algorithm is you can use a bunch of them to classify a random sequence and decide where it came from, which could be useful in a code-breaking attempt. So my genetic algorithm, you start with the population initialization phase. I chose to represent the genomes as trees of numbers, and I'll explain how these translate into computer programs in a minute, but each number in this tree is called a node. Take that 17, for example. Its parent node is 2, its children nodes are 0 and 10, and its sibling node is 3. And the nodes are linked like this to form a tree of numbers, and each one is a genome. So the objective function will then evaluate these trees as programs and decide what the output is and how good that output is. So it takes the population of genomes, 60 of them, which were created in the population initialization phase, and the objective function runs, and it produces a list of scores. So the first step in the objective function is to translate a tree of numbers into a computer program. And each node, each number that is a valid number in the tree, which happens to be between 0 and 18, corresponds to an operator. So every node that has children nodes, not the ones on the bottom, is then translated into an operator which performs a specific function on inputs and gives an output. And so let's examine the plus operator and see how it works. This plus operator takes input from the nodes below it, 8, 4, and 2 in this case. And because it's an addition operator, it will send on the sum, 14, to any node above it, which in the tree represented there was a while. So the while would then take in the 14 as input from the node below it. And there are some special operators which do things that you might not expect. The addition, subtraction, multiplication, and division are pretty self-explanatory. But this if operator will take in a 0 or a 1, or any number on its second input. If that number is a 0, then it will pass on a 0 to the next node above it. If that number is not 0, 1 for example, then it will pass on what it received on the first node to the node above it. That allows it to implement a common control structure in computer programming. While is another common structure. This, what a while does in this case is it will take in a number on its second input. If that number is a 1, then it will go back and repeat everything below it until that number is a 0, at which point it will pass on what it has on the first input. And that's not very useful unless something changes below it each time you run. So the accumulator operator exists. And what that does is it takes in a number and it totals that up with everything it has received before, and it sends that total upward. So as you run this accumulator operator multiple times, it would produce successively larger values. And the input operator is the most important in the tree. The input operator will take a value from below, and it will allow the program being run to take in a value from the random sequence that it's operating on. For example, this input operator takes 7 as input, so it will look at the random sequence, which is pictured to the left, and it will find the 7th number and pass that on as output, in this case 36. So once you've got a program with a bunch of operators that are defined, each operating on the nodes below it and passing on the resulting value to the top, the root node operates on its inputs and produces a value, and that's the return value of the tree. That is what is passed on as output from the tree to decide whether a sequence is random or not, whether it's from one source or another. So once you've got a program that you can evaluate, the objective function goes and takes 128 sequences from one source of random numbers and 128 sequences from another source, and it will push them through the program, and it will produce one value per random sequence. Then it will perform what's called an ROC analysis. And an ROC analysis is a way of deciding where you should draw a divider line to separate a bunch of numbers into two groups. So the computer does this a bit differently internally, but if you were to do it by hand, you'd draw a frequency plot of the data. For example, the red line there could represent the frequency of the results from the random sequences being pushed through the genetic algorithm, being pushed through the program under development. Along the x-axis, you have a result, and on the y-axis, a frequency. And the green line could represent the results from when you push in a sequence from source 2. And so it then decides on the best dividing line. And then it computes what's called the sensitivity and the specificity. The sensitivity is where you take all of the numbers that come from one source that lie on one side of the line and divide it by the total number of all results from that source. So you come up with a fraction between 1/2 and 1. And the specificity is the same thing, but for the other data set. You have the total number of points which are on the other side of the line from the other source, and you divide it by the total number of points from that source, coming up with another fraction between 1/2 and 1. And the sensitivity and specificity are both things that you want to increase, because as they increase, those two groups move apart, and the program is better able to decide where a certain sequence of numbers came from. So the sensitivity plus the specificity equals the objective score. And the objective score is the measure of how good a certain program is. So once you've decided on an objective score, you go on and put the population in order. So if you have a bunch of genomes in the population with objective scores, they're put in order, and then the bottom 20 genomes, in reality the population is 60, but due to lack of drawing time I only drew 6, removes the bottom third of the population, and then it moves on to the breeding phase. The breeding phase is when it takes the top third of the population and breeds those genomes to produce new children. And to breed two genomes, you take both genomes and you duplicate them. So if these are the duplicates, these will eventually become the children. Then you select one node in each genome, and you swap those nodes and everything below them, so that you now end up with 2 children. And you repeat that node swapping process several times, and you come up with a bunch of children which are similar to their parents and descended from them. So once you've done that, then it occasionally mutates one of the children. So here's one of the children that was just bred, and it will select 2 nodes within that child and swap them. And then it will insert these new children into the population to replace the ones that were killed off. And then the cycle will repeat for 1,000 generations. I just picked 1,000 because it seems like a number which didn't take too long and still produced reasonable results. And after 1,000 generations, the best genome would be selected as the final result. Now, the final result can be one of 2 things. It can be a general solution, or it can be a case-based solution. A general solution is one where the program will decide where a certain random sequence came from based on actual qualities that are inherent in sequences from one source or the other or both. But a case-based solution is one that merely operates on coincidences that occurred in the random sequences that it was tested with. And to understand why a case-based solution occurs, I used a real random number generator to generate these 100 random numbers, and I divided them into 2 groups of 5 sequences of 10 numbers each. And I looked for a pattern in here which could be used to differentiate these 2 groups of sequences, which all came from the same source. And I found that if you take the second number in the sequence, if it's less than 100, it comes from the first group. If it's greater than 100, it comes from the second group. And this rule works 80% of the time. So it's very simple to come up with a case-based solution which has absolutely nothing to do with the qualities that are actually present in random sequences from a given source. And once you've got so to test this program to see whether it's a general solution or a case-based solution, I took another complete set of random sequences and ran the objective function one more time. If its score stayed relatively constant, it was a general solution. If it fell significantly, then it was a case-based solution because it suddenly lost all the coincidences that it was working with. So my results. Unfortunately, I didn't get any spectacular results, but I was able to differentiate between sequences from a linear congruential generator and real random data one time in 63 attempts with a 1.58% success rate. And I was able to differentiate between sequences from linear congruential generators and the rand function in my computer, which is a pseudorandom number generator, known to be pretty good, 4 times out of 40 attempts, or 10% of the time. So while this result is not an incredible result, it does show that this technique for differentiating random sequences is a viable one that merits further work. And the other solutions which were arrived at were either low-scoring, case-based, or both, meaning that the genetic algorithm did not successfully come up with a method of differentiating between the two sources. So this is the evolutionary progress of a case-based solution. And in a case-based solution, along the bottom is the generation number, so that tracks the evolutionary progress. And on the y-axis is the objective score. And as you can see, there was an initial period of stagnation as the randomly generated solutions got up to speed with the best member of the population. Then there was a period of rapid growth, tapering off, and the development basically halted at around 1.5 or 1.6. The evolution of a general solution, however, is far more interesting. It began as a case-based solution, but eventually, at around generation 570, it discovered the key to the problem and decided that it had, and discovered what was necessary to differentiate between the two sources that were used to develop this program that was under development. And it rapidly grew in its ability to differentiate random sequences until it had reached the maximum score of 2. And I've attempted to improve the genetic algorithm several ways, and none of these improvements that I've attempted has come up with any great results improving the genetic algorithm. But there's still further work to be done, and probably the most important thing will be to consult with someone who actually knows what they're doing with genetic algorithms. I was mostly working on my own, but it would be a great help to me if I could consult with someone who has experience with genetic algorithms. And some potential applications of the genetic algorithm are using the genetic algorithm to decide whether a certain pseudorandom generator is of good quality or not, and using the sequences produced to decide or discover where a certain sequence of random numbers came from. And also, I recently realized that this could also be beneficial to projects like SETI, the Search for Extraterrestrial Intelligence, where they're trying to find signals in the cosmic rays they receive to see if they can find signals of alien life elsewhere in the universe. And this could help them decide whether they're receiving signals that are non-random or not. It could assist projects like that. So, any questions? Yep, Max?
How long did it take to run the 1,000 generations?
It usually took about 20 minutes, depending on what version of the genetic algorithm I was trying. Sometimes I had to cut it off at 1 hour, but it usually completed 1,000 generations in about 20 minutes.
Academy, and Matt won first place at Academy.
Thank you. So in 1969, the salt marsh in Wild Harbor was contaminated by number 2 fuel oil spilled by the oil barge Florida. As you can see in this figure, the prevailing winds drove most of the oil into the Wild Harbor salt marsh, where the effects were swift. The mass death of both benthic organisms and salt marsh grasses occurred soon after the oil spill. 30 years after the spill, Reddy and others found the presence of petroleum hydrocarbons 12 to 16 centimeters below the marsh surface. So my project studied the effect of this continued presence of hydrocarbons upon the marsh sediment. So one of the plants most affected by the oil spill was Spartina alterniflora, a common New England salt marsh plant that grows close to the tidal channel. So in 2002, Lynn and others showed that the number 2 fuel oil, which was the oil spilled during the oil spill, reduces the below-ground biomass of Spartina alterniflora significantly, especially the roots, as opposed to the rhizomes. So, and in fact, according to a recent study, areas in Wild Harbor that were more contaminated during the oil spill have less below-ground biomass today than areas that were less contaminated. So the significance in this reduction of below-ground biomass lies in the fact that the roots and rhizomes of Spartina alterniflora are good at trapping sediment, increasing sediment accretion and decreasing sediment erosion. So if oil decreases the below-ground biomass, it causes the marsh sediments to become loose and unstable, more susceptible to increased redistribution by tides and storms, and increased overall erosion. So the purpose of this project was to determine how the presence of petroleum hydrocarbons influences salt marsh sediments in Wild Harbor, in order to better understand the long-term effects of oil spills on coastal salt marsh ecosystems. And there are 2 stages to the project. The first stage investigated whether the presence of petroleum hydrocarbons influences salt marsh topography by examining the differences in topography between an oiled and a non-oiled site in Wild Harbor. The second stage investigated whether differences in below-ground biomass would account for variation in elevation observed during stage 1. You see, I knew that the oiled site had less below-ground biomass than the non-oiled site, and I knew from stage 1 that the oiled site had a higher variation in elevation. So what I wanted to establish was a link between these two, to show that below-ground biomass truly does affect elevation. So to do this, I compared the below-ground biomass levels of stable and eroded areas in the oiled site alone. So as for the hypothesis of stage 1, it was hypothesized that the oil would cause the loose sediment, and that would cause the uneven redistribution by tides and storms and the increased erosion. So what I expected is that these would cause, respectively, a greater variation in elevation at the oiled site and a lower marsh surface at the oiled site. So as for the methods, the 2 sites in Wild Harbor were chosen close together, about a quarter kilometer apart, to reduce the variability in conditions subject to location. The oiled site was chosen where the concentration of persistent petroleum hydrocarbons is very high, while the non-oiled site received relatively little oil during the oil spill, and the concentration there is negligible. So to determine differences in topography between the 2 sites, I measured the elevation of each meter along radial lines from a center point. So in this figure, at each of those dots, I measured the elevation relative to the center point, and using the water equilibrium method, which I'll explain in a minute. As you can see, the data points are more concentrated in the lower semicircle of each site, because that's where I expected the most variation in elevation. So the next stage was to determine the elevation difference between the center points of the 2 sites, so I could accurately compare the 2 sites to each other instead of just comparing areas within the sites themselves. So to do this, I compared the tide levels at the sites after the same high tide, and after this I corrected the elevation values of the non-oiled site so that all the elevation values were relative to the same thing, the center point of the oiled site. So before I move on, I'll quickly explain the water equilibrium method. So this method required a clear plastic carboy, or plastic jug, and attached clear vinyl tubing, and all that filled with water. And the unattached end of the tubing was attached to a meter stick,
so that to facilitate the reading of the water level. So each time I filled the apparatus with water, I brought the unattached end of the tubing to the center point of each site and measured the water level there. Then, when I brought the tubing to different points, or data points, I measured the water level there. So then I can compare the water level there to the water level at the center point, which would give me the elevation of that data point relative to the center point. So as for stage 2, it was hypothesized that because below-ground biomass is so good at trapping sediment, stable areas of the oiled site would have higher below-ground biomass levels than eroded areas. And as for the methods, 5 sites were chosen in the oiled site where adjacent points, the same distance from the center, had large differences in elevation. And so at each of these sites, I chose 2 areas, one at a high elevation, presumably stable, and one at a low elevation, presumably eroded, and sampled them for below-ground biomass by removing a 19 to 20 centimeter core. And to do this, well, I removed the core, and then I sorted the biomass of the core into live roots, live rhizomes, and dead matter before the measurement of the mass. And you can see the sorting process in this picture here. So now for the results. So in analyzing the elevation data for stage 1, all the elevation values are relative to the same thing, the center point of the oiled site. Also, I analyzed only the lowest semicircle of each site, which, remember, had the highest concentration of data points. So this graph shows the general topography of the oiled and non-oiled sites. And there are 2 main differences between the 2 sites visible in these figures. First of all, the oiled site had many more pits and channels, contributing to an irregular marked surface. On the other hand, the non-oiled site had very few pits and channels, and in fact had a very evenly graded slope before the tidal channel, with the exception of this island. And secondly, the second difference between the 2 sites visible in these figures was the higher elevation of the upper section of the non-oiled site. You see a lot of darker reds in the non-oiled site that you don't see in the oiled site. So the next step was to quantify these results. And the first way I did this was to analyze elevation of arcs of points, the same distance from the center. So you would expect these points to have similar elevations based on the roughly semicircular shape of each site. So the first thing I found was the mean relative elevation at each distance from the center, with the oiled site in blue and the non-oiled site in red. And this graph clearly shows the higher elevation of the non-oiled site surface. The second thing I did, again analyzing along arcs of points, was to find the standard error of elevation at each distance from the center. And again, the oiled site is blue and the non-oiled site is red. So standard error is a measure of the variability of points, taking into account sample size. So a high standard error would represent a high variation in elevation. So you can see that at each distance from the center, the oiled site had a higher standard error than the non-oiled site, showing that it had a higher variation in elevation. The next way I analyzed the elevation data was to analyze elevation data along radial lines from the center point. So for each of these radial lines, I calculated a 3-point running mean for the first 7 points, which can be seen here in red. The actual data is shown in blue. So what this, in effect, creates is a smooth version of each radial line. So, and what I looked at was the deviation between, or difference between the real data and the running mean. So if this distance is very high, it means there was a very high variation in elevation. So what this graph actually is the histogram of those deviations from the running mean. And again, the oiled site is blue and the non-oiled site is red. So, and the frequency is on the y-axis, and the deviation from the running mean in bins is on the x-axis. So the first difference you see is that the non-oiled site had a much higher frequency of low deviations than the oiled site, showing that it had a very low variation in elevation. Also, the oiled site retained some medium frequencies of high deviations, showing that it had a high variation in elevation. So, and a chi-square test determined that there was a statistical difference between the frequencies of the 2 sites. So now for stage 2. Remember that the data were only comparable in pairs, because only for each pair of stable and eroded areas was it assumed that they should be at the same elevation. So what this graph actually shows is the dry below ground biomass of the eroded areas on the y-axis plotted against the dry below ground biomass of the stable areas. And a one-to-one line is plotted. And it's plotted both in terms of live roots, brown, and live rhizomes, green. So you can see that if a data point lies below this one-to-one line, it means the dry below ground biomass of the stable areas was greater than that of the eroded areas, which is what I expected. As you can see, all of the data points lie below the one-to-one line. So in sum, the results of this project supported the hypothesis. The null hypothesis that there was no differences between the two sites was rejected. In stage 1, there were 2 topographical differences supported by both qualitative and quantitative data. And they were a higher variation in elevation at the oil site and a lower marsh surface at the oil site. In stage 2, it was shown that the stable areas had higher below ground biomass levels than the eroded areas. So before moving on to the significance of this, I'll quickly go over the sources of error for the experiment. First of all, the differences in topography might have been caused simply by the differences in location, although I minimized this by placing the sites very close together, about a quarter kilometer apart. Secondly, the elevation of the center point might have sunk during the course of the experiment due to trampling, although I minimized this by placing the center points in relatively stable areas. And finally, during stage 2, different levels of compression of the sediment could have occurred during the core removal, which would have influenced the below ground biomass level. So taken all together, the results of stage 1 suggest that the sediment at the oil site is much looser than that at the non-oil site. And the logic behind this is that loose sediment would cause an increased redistribution by tides and storms and increased overall erosion, which would account for, respectively, the increased variation in elevation at the oil site and the lower marsh surface at the oil site. And stage 2, if you remember, was designed to determine how the presence of oil made this marsh sediment loose. And remember that I knew that the oil site had less below ground biomass, and I knew that it had a higher variation in elevation. So I wanted to know that since the oil decreased the below ground biomass, as you can see in the flow chart, would this actually cause the loose sediments? And according to my experiment, the answer was yes, because the below ground biomass was shown to counteract erosion. So I can complete this flow chart, which then leaves me back up to the stage 1 results. So the overall conclusion of the experiment was that oil spills likely affect coastal salt marsh topography for decades after contamination. And as for future directions, I could investigate the ecological consequences of the differences between the two sites by mapping plant species distribution over a series of years. And plant species distribution is largely dependent upon elevation, which is in turn dependent upon erosion. So if the erosion rates are different at the two sites, there should be some ecological differences between the two sites. And to conclude, I would like to thank a number of people, but especially my mentor, Jen Culbertson. Thank you.
How did you insert your water tube apparatus into the ground without disturbing your readings? Your diagram showed it below the surface.
Oh, it's not below the surface. I'll show you the diagram again. It's not confusing.
I told him he had to change it.
Let's see. It's just this perspective. It's on the surface.
Ah, I see.
It's not below the ground.
That makes more sense. Thanks.
So I have a question for you, since I'm one of Jen Culbertson's mentors.
Yeah.
She wrote, I think, 5 times
Yeah.
last week. She had a nice article about some of her work on fiddler crabs. Fiddler crabs were one of the key species impacted in the few years directly after the spill, because she finds that the population fell back, the birds, feeding behavior
the tubes.
Not as deep and they're not as extensive as they would in an unoiled marsh.
I don't think it significantly affects the topography because they're just small holes and they're mostly near the bank as opposed to actually where the grass grows the most.
How about the mussels that she works on? If they would be, near the marsh grasses.
Actually, they're actually close to the marsh shore too. I think both of those things, they're definitely affected by the oil spills, but I don't think they have, I think they have a small influence on the actual topography of the marsh.
One is the level at A and B are the same.
Yep.
Why isn't the water in the jug far away at the same level?
Why what?
Why isn't the fluid in carboy at the same level as A and B?
It would be. It is.
Isn't it?
Yes, it is.
Perspective drum.
I see. Perspective, right. Next question is, it seems reasonable to me, the conclusion you came to, because oil does make things slippery, and allows for more rapid erosion.
Well, I think the oil is only slippery in the first couple weeks after the oil spill, and then it sinks down into the sediment where it's not slippery anymore.
And it gets tied up.
Then it gets tied up in the sediment,
And
and
it
well, it loosens it by hurting the plant roots, and the plant roots hold the soil together. So that's how it loosens it, rather than it being slippery.
I see,
Yeah, nature doesn't always give you a perfect situation. Could the wind direction relative to where your site is have a significant influence on the amount of erosion if the two were actually identical or something like that?
Yeah, the wind and the tides and all that would have influence on the topography. But I tried to make the sites are very close together, so I assume they are influenced by the same tides and the same wind.
Well, each of the speakers will have their poster boards up here. Come and talk to them. I'd like to congratulate all of them for their really outstanding projects, and I wish them luck in the state fair, the international fair, wherever they go on from here. So join me in congratulating them.